Zara already did brief introductions for those who were here, but for those who weren't, Dr. Mostaço Guidolin is actually an alumnus of the BME program. I believe she originally did her undergrad in medical physics in Brazil at the University of Sao Paulo, and then moved here where she did her PhD with Dr. Major and Dr. Ko, looking at various image processing methods related to heart imaging and things like that. Graduated with her PhD, I believe in 2015,
Yeah.
and then went to UBC to do a postdoc with, I was just talking with Zara before the seminar, with the very first speaker of the BME seminar series. So that was actually even before I was here, but it sounded like these are good opportunities to meet people within your field, and whenever you're at conferences and things like that, don't be shy to mingle with like big name people in your field, because that's how you make connections. So ended up going to do a postdoc in a very prestigious lab, has won a number of awards since she's been there, including I believe most recently a CIHR postdoctoral fellowship, several presentation awards and things like that. And then we'll actually be moving to Ottawa to start a faculty position at Carleton University in January. And so very excited to welcome you back and to hear about some of the work you've been doing lately and some of the work you'll be doing in Ottawa.
Thank you very much. So I'm sorry for all the problems here we have, but with Max we never know what's going to happen. So yeah, today I'm going to start with almost a picture of myself. That's how I feel with the work I do. It's since my undergrad, I have been always working the interface between biology, medicine, and engineering and physics. I have worked with a bunch of different methods, optical methods, microscopy, spectroscopy, always trying to characterize biological tissue. I have worked with cells characterization, like Dr. Shep mentioned, I did my PhD characterizing cardiovascular tissue, and most recently I have been working with respiratory diseases. And I have been shifting slowly into collagen characterization, and that's what I would like to share with you today. It's the work that I have done in the past almost 4 years now. So today what I will tell you is that I will present to you who are not familiar with optical techniques. I will present to you nonlinear optical microscopy, and I will tell you how powerful and how nice this technique is for imaging collagen and elastin. I'll also tell you how we can use those types of images to get some very interesting information for characterizing, in this case, asthmatic areas or tissue in general. And then I will finish my presentation sharing with you a little bit of my plans as I move to Ottawa and I start my lab in combining imaging techniques and 3D bioprinting. So that's my future goals kind of thing. So yeah, so let's start talking. I would like to share with you why collagen is such an important molecule when we are talking about biological tissues. Collagen is a very special molecule, a very special protein that it's only animals produce, and over 25% of all animals' composition is made of collagen. So it's a very nice and interesting molecule that we should be keeping track of, because collagen has a very unique structure. So we have usually, sorry, I don't have anything but we have those combinations of some amino acids forming what's called the alpha chains. And each one of those chains, they get organized in a way that forms the fibrils. And fibrils will form then the collagen fibers. So if you imagine a very thick rope, we have that big rope, and inside we will see kind of the threads organized to form that big rope. So that's the same idea with collagen. We have all these structures going all the way from the fibers all the way down to the alpha chains. And these proteins, they are very strong. And that's why they are so important for the composition of tissue and the animals in general. We can compare collagen to they have the tensile strength similar to a steel cable. So from there you can imagine how good they are for structural things. And it's also important to know that collagen, we have 21 types of collagen there. And some of them are fibrillar and some of them are non-fibrillar. So that means some form these ropes and some not. So that's an overview of why collagen is so cool. And when we are talking about diseases or tissue in general, we can see from these examples, those are images from my PhD actually, and they are all from an artery. So we can see the variety of the collagen shapes that we can get in there, even if we are looking at the same type of tissue and the same type of disease. So we can see a lot of changes happening with collagen. And that's something that's really interesting and important to be studied if you want to understand what's going on with several diseases. Usually, in many organs, in many areas of the body, we will have some changes that eventually will get back associated to collagen. If we talk about cancer, we will have accumulation of more or less collagen there. If we talk about fibrosis, atherosclerosis, gum disorders, skin disorders, even if you cut your hand and you have a scar, that scar is going to be made of collagen being accumulated there to heal the tissue. So today I would like to talk a little bit more about asthma, which has been the disease that I have been working most recently. And the very first person to define asthma was Dr. Salter. He was a British researcher, and when he was a kid, he had asthma. And eventually, as he grew up and he became a scientist, he made the point of his life to study and understand asthma. What was going on? He wanted to know better why he couldn't breathe, why everyone had a difficulty there. And he was responsible for writing the very first complete manuscript or book about asthma. So that it was the gold standard for many, many years. And he was also the first one to image an airway, an asthmatic airway. So that's the very first picture of a lung, an asthmatic lung. And it's from 1864. And we see here where the arrows are going, where the two airways are. So the image is not that nice, but for 1864, it's actually pretty good. Whoops. Then we move forward a little bit. 65 years later, we had Dr. Collagen coming along. And he was the one who developed this kind of histological staining. So it's called the Masson's trichrome stain. And that was a revolution at that time, because for the very first time, it was possible to visualize different components of the tissue. So here in this example, we can see some structures in reddish, some a little more blue. So that was a very novel way to actually see all the structures and all the changes that were happening. If we move a little more, today, 2019, 90 years later, after the development of that stain, that's how we still look at the tissue. So we still rely on chemical stains, we're still trying to get information from the tissue based on stains. I don't have to say that. We should expect to see something a little more up to date, right, at this point. And although it's still a very good way to visualize many of these structures, as you can see here, we don't see the fibrillar information. We don't see the fibers. We see different colors, we see structures, but we don't see what's in the tissue with a higher resolution per se. So, for example, in the case of collagen, which is the focus of my talk today, we have a few possibilities for visualizing those proteins. I'm sorry if the slides are a little bit weird there. It's just because I had to export from the Mac to PowerPoint, so some things are a little shifted. But anyway, so for example, if we use the Masson's trichrome, we can see the collagen there in blue. If we use another type of staining, we can see collagen in red, in pink. Or if we want to see elastin, we can use a different kind of stain, so we see elastin in brownish. But it's all very time consuming. We have to prepare the sample, we have to do a bunch of steps for chemically staining all this tissue. And that's not necessarily the best way to do it. So in the case of asthma, when we compare an asthmatic airway from one normal control patient with an asthmatic one, we can see the changes are really dramatic. The main features that I have been looking at during these past years were related to the increasing of collagen here in this region, the lamina propria region. So everything that's in blue there, it's basically collagen being accumulated. So as someone has asthma, we can see what's called remodeling. So instead of having a nice and open structures that we see on the left, we start to see a lot of thickening around the wall. We can see mucus being accumulated. We can see the epithelium here getting a little thicker. So all these changes we can visualize using stains, but we don't have information about how these collagen fibers, for example, are changing there. And collagen is one of the main components also of the extracellular matrix. And the extracellular matrix is the network that we have basically everywhere in the tissue, and that will help the cells to move around, to get the responses, the chemical responses. So the structure of this extracellular matrix is very important for keeping everything in equilibrium, in a way that all the responses, the biological responses happen the way they should happen. If we start changing a little bit how the extracellular matrix is composed, we start to have a little problem. And one very interesting technique to visualize how these changes in the matrix in collagen tissue that I have been using the past few years is called second harmonic generation. That's a type of microscopy. And the main difference between a regular wide field microscopy and second harmonic is that we use a laser as a light source instead of just a regular light bulb. So we have the laser, we shine this laser into the sample. Depending on the composition of the sample, sometimes we will have this phenomenon called second harmonics. And that's basically we will have a wave of a different color coming from the sample that will have 2 times the original frequency of the laser. And in the case of biological samples, collagen is one of those materials. So if we shine laser into something that has collagen, we will get this special light coming from there without the need of any staining. Another technique that I have used quite a bit is called 2-photon excitation fluorescence. That's very similar to the regular fluorescence that in many biology labs they use. The difference here is instead of, again, using a regular laser source, or instead of using 1 photon to generate the fluorescence, we will have to have 2 photons being combined at the same place at the same time, and then what we will detect is the signal that those 2 photons will generate. The advantage of this kind of microscopy is that we don't have too much background, we don't have signal coming from all over the place. We will have the signal just coming from that very specific place where the 2 photons got combined. So here on the right side, I have some examples just to show you how nicer those images can look. So in the very top, we have an example of a wide field microscopy, fluorescence. Then in the middle is what we could get using confocal microscopy. It's much better. But then if we do the 2-photon fluorescence, we can see the level of detail, and the spatial resolution is so much clearer that we can see all the distortions in a much better way. So going back to the original problem, how can we see better what's going on inside of the asthmatic area? Before we had the stains, it's nice, we can see the structure, we can see all the types of tissue. But if we use nonlinear optical microscopy, we can see, for example, collagen and elastin with a much better level of detail than we could otherwise see using chemical stains. So here I would like you to pay attention how nicely we can see the fibrillar, all the fibers with a little better resolution than we could see before. And one very interesting thing is for getting those images using nonlinear microscopy, we don't need to stain the samples. So all the signal is coming from whatever structure is in there already. So you don't have to prepare the tissue, you don't have to do anything. It's basically you just put under the microscope and you get this kind of images. So the question I have been working in the past few years has been related to, okay, so we have this very cool microscopy technique. We would like to know a little better what's going on with collagen and elastin. And how do you explain, what's their role in when the remodeling happens in asthma? So the hypothesis I have been working with was that the fibrillar collagen in asthma, it's more fragmented because of defective repair that happens with asthma patients. Okay, so we had all these nice images and everything, but I had to find a way to actually quantify what we could see. I could not just show, okay, we have more fibers, less fibers, whatever. And the way I have been trying to quantitatively address or try to track those changes in asthma has been based on texture analysis. So I'm not sure how many of you are familiar with imaging processing, but I will just explain very quickly. So usually we have an image. The image is made of several pixels. The pixels have intensities, which are basically the colors or the gray levels that we see there. And those colors or gray levels, they are numbers. So we can do several mathematical operations with these numbers. We can calculate, for example, the average, we can calculate the standard deviation, we can calculate the kurtosis, and so on. So the intensity that we get in an image is directly related to the amount of certain components that we are imaging. So for example, let's go back to the collagen idea. If I do the second harmonic microscopy, the amount of signal I will be getting there, it's going to be related to the amount of collagen that I have in the sample that I'm imaging. However, sometimes we have problems like this or cases like this. So in this case, we have 3 different images. We can clearly see they are different. But if we try to calculate, for example, the mean, we will get the same number because the number of black and white pixels is the same. But a way to get around it and try to actually quantify these differences is based on what's called gray level concurrence matrix. I won't go into too much details here, but based on the intensity of the pixels, we can do some calculations, and then we can find a relationship between the neighbors, all the neighbors of each pixel. And therefore, we can track the structural changes, or we can see the morphology of the objects that you are imaging. We can possibly calculate, based on this set of textures, we can have 14 different parameters. But today I will just focus on 2 of them. So for example, we have the entropy. Just like in physics, entropy is a measure of how disorganized things are. So if we try to calculate the entropy of an image, going back to our example, we will be able to distinguish those. So before we had the mean, could not see any difference, but if we move on to these textural features or the gray level features, we can eventually see these differences. And for this work related to asthma, I have been calculating those textural parameters. And we started to look what was happening inside of the large areas. I started to calculate what's the entropy, what's the inverse difference moment of these images or the collagen, and try to see if we could see any difference between the controls and the asthma ones. What we could notice was we have the intense one is telling us how much collagen or how much elastin we have in each case. So we could see we have more collagen in the case of asthmas, but that was not big news. We could see that already with the chemical stains. However, when we calculated the entropy, that was something very interesting, especially for the biologists. Because for the first time, we managed to show that those collagen fibers, they were actually more disorganized than in asthma than they were in controls. And when we move on, we calculated another feature called IBM. That one is telling us the level of fragmentation of these fibers. So you can see that the asthma one is a little less fragmented, it's a little more fragmented than the control one. For the elastin, we couldn't see much of a difference when comparing the 2 cases. And that was the point when we decided, okay, we are looking at the extracellular matrix, but since the elastin is not showing any obvious changes, let's focus on what's going on with the collagen. So large airways is everything that has 2 or more millimeters in diameter. So that includes the trachea and the bronchi. But we also decided, okay, but let's take a look if there is anything happening in the smaller ones, the bronchioles. So anything that's smaller than 2 millimeters, it's called, by definition in the area, it's called small airways. And that's something that we also took a look using linear microscopy. So again, it was interesting because we could see the same pattern happening in both, from the trachea all the way down to the bronchioles in the lung. We had more collagen being accumulated when we looked at the asthma airways. We saw the collagen fibers being a little more disorganized. We saw them being more fragmented, not with changes in elastin. We also looked into the orientation of these fibers, because that's another parameter we can look and see if anything was changing. So for the controls, here on the top, we can see very clearly that the collagen fibers are very well organized. They are very nice fibers. And as we look at the asthmatic ones, we just see lots of fragments, not necessarily oriented in a specific direction. And that's also another parameter that we could obtain for both the small and the large areas. And finally, we also looked into how these fibers were changing in terms of, first of all, the thickness. We were measuring how thick each one were for both asthma controls, the small and large airways. We couldn't see any difference there. But then eventually, we started to measure how far apart they were, what's the spacing between them. And that's where we found some changes. We found that in asthma, those collagen fibers were a little more spaced than in controls. And that was something we started to wonder, okay, so what's going on there? So up to now, we can see that the nonlinear is a really powerful tool to characterize the extracellular structure. We got to know that the fibrillar collagen is more disorganized in asthma. We couldn't find any changes related to sex, age, and even cases of fatal asthma. And the elastin did not change too much. So at that point, we started to wonder, okay, so now we need to understand a little bit better what's causing those changes, from where those changes are coming from, which cellular mechanisms are causing that. So then we started to combine, we went back a little bit, started to combine some focal microscopy with second harmonic. So I have been brainwashing you to think, oops, that second harmonic is always the blue stuff, but only for this image, the collagen, the second harmonic is purple, so only for this one. So here we stained actually some cells. The nuclei of all the cells are showing red. We also stained the cytoskeleton of some mesenchymal cells, which is shown green. And then the collagen are showing purple there. We could see that in controls, we have nice bands of the smooth muscle mass. We could see the band of collagen, the cells over there, the epithelium over the nice. But as we go and look at the asthma, we don't have that pattern or that organized structure anymore. So we see the cells are migrating, all the green spots here are migrating into the tissue. We see collagen basically everywhere. So that led us to think, okay, so maybe we should start to look into the fibroblasts, because the fibroblasts are the cells responsible for making collagen. So maybe they are the ones that are causing all these problems regarding the collagen disorganization and the matrix changes that are happening in asthma. So to test how these fibroblasts were working, we got from the same patients, from the same donors that we were doing the images, the nonlinear images, we got cells, and then we did some in vitro assays, which is, we basically had collagen gels. We seeded those fibroblasts from both controls and asthmatic people, and we waited to see if those fibroblasts would eventually make fibers off of this collagen that was in there. If they were working fine, we should see a lot of nice fibrillar collagen. If not, we wouldn't see any of them. So the very first thing we could notice was, after 72 hours, the gel was contracted. So that means the cells were actually making those fibers and pulling all these structures, so making the gel a little smaller. And when we compared the percentage of contraction of the controls and the asthma, we could see that the asthmatic ones, the cells were not able to contract or to make fibers as nicely as the control ones. And then to double check that, we used again the second harmonic microscopy, which was good to see fibrillar structures or collagen fibers. And again, we could see a lot of fibers made from the controls fibroblasts and not so many from the asthmatics. So that was telling us, okay, so the asthmatic cells, they are not working properly, making the collagen or remodeling the collagen. There is something going on there. And that's our mini conclusion. So, okay, so all the fibroblasts related to asthma, they are defective at this collagen contraction. But that was not enough. So we started to go down and see, okay, but what's wrong with these cells? So we tested several parameters, or we tested several proteins, or RNA expressions, and so on. And we looked at for example, the cell stiffness, the distance between the cells to see if there was any difference between them. We tested if the cytoskeleton of the cells were working properly, because depending on, here it's just a diagram of those cells. So depending on these proteins, or depending how they are arranged or how they are expressed, the cell might be able to migrate better, they might be able to do their job properly, and we couldn't find any difference. So the cells, as you look at them, or from the point of view of the protein expression, we didn't notice any changes. So we were like, okay, there is something wrong, but we don't know yet what was there. So the mini conclusion, number 2, was that all the fibroblasts from asthma, they actually had a normal functional cytoskeleton. And that was a little surprising. But eventually, we decided to test one last suspect, which was decorin. Decorin is a protein that's responsible, the name comes from, because it decorates the collagen fibers. So we have the collagen and we have this decorin all attached to the fibers. And it plays a role in the assembling of this matrix. And also it's very important for making sure that the spacing between the fibrils are somehow consistent or properly. And I don't know if you remember, but I told before that we found some difference in spacing regarding the collagen. So that's what led us to test this last suspect. So, yeah, so when we tested, we did some experiments to test if the expression of decorin was different between controls and asthma. We found that the asthma ones were expressing less decorin, so therefore they were not properly spaced, they should not be properly spacing these fibrils inside of the fibers, and therefore probably that was the cause of all these changes that we were seeing in the matrix. So in order to test that, we decided to go and image those collagen fibers with a different technique. So up to now we have been dealing with molecular microscopy, looking at the fibers. Then, after we saw this change in decorin, we decided, okay, so let's take a look using transmission electron microscopy and see if we can find anything inside of these collagen fibers, so at the fibril level. And that's an example, representative image of what we found when we looked inside of the collagen fiber. So the control ones, we can see all the structures somehow very well organized, and the asthmatic, okay. And then, doing some processing, we could quantify, for example, the circularity degree, which is basically how close to a perfect circle each one of those structures were. When we look at the asthma, they are much more circular-like than the control ones. We also could see that there is a much bigger variety of structures in there when compared to the controls. So the conclusion that we got from this work was that yes, all the asthma-derived fibroblasts were defective at packing collagen because of lower decorin expression. So that was great. Very interesting, very exciting. That has been my work for the past almost 4 years now. But one thing that I have been very intrigued by was, okay, it's all very nice, but we have such a variety of tissues, organs, arrangements, and I always wanted to try to push the boundary a little more. And that's where I got myself into 3D bioprinting. And that's where I'm planning to move forward as I go to my new position. So usually what we do, we have cell cultures. We have those models and ways to try to mimic what's going on in the tissue. But we can see that it's a very different arrangement of what we find in the real tissue. So in the real environment, in the natural natural environment. The cells are all able to move in a three-dimensional direction. They are not blocked by basically, let's say, the glass or the floor of the dish, the bottom of the dish. So that's affecting a lot how the cells behave. So for example here, that's a diagram showing an artistic representation of a tumor. And that's how we usually look at how we do the experiments relating to tumors and stuff like that. So we have a cell culture, we see what they are doing, and we try to draw conclusions. However, if we try to print something or work with organoids, we can try to assemble something that's a little closer to what we see in nature than what we have been doing up to now. So that's my biggest motivation, many others' motivation, to move towards trying to basically redo all these experiments in an environment closer to what we observe in the real world. So one big focus of my future research will be fibrosis. So fibrosis, just quickly, it's related to the accumulation of collagen, among other things, into several tissues. And what is very interesting for me in terms of fibrosis is it's not related to one specific disease. That happens in so many cases or pathologies that it's going to be really interesting to see what's going on there. And in the case of asthma is a disease that has no cure yet. So we have the possible treatments just to alleviate the symptoms, but there is no way to cure that yet. And part of it is because we are targeting the inflammatory responses. But not necessarily that's the solution for the problem. There is evidence that some of the mechanisms that drive fibrosis, they are distinct from those targeting inflammation. So in asthma, we have both happening. We have inflammation and we have fibrosis. And we are only looking at the inflammatory side of it. And also there is a little bit of evidence showing that the inflammation is needed to reverse the fibrosis. So it's a little bit counterintuitive, but the reality is we don't know what's going on there. And the way the fibroblasts behave, it can be affected by so many things, but by the chemicals, by the chemical responses that we have in the tissue, but it can also be affected by how the matrix or how the mechanical environment is there. So the million-dollar question these days for asthma is, what's coming first? Is that the fibrosis that's causing the inflammation, or the inflammation is driving the fibrosis? And that's the big question that is preventing us to find a cure for asthma. So which one we should target first when we think about developing drugs? We should target the collagen deposition, the fibrosis, or we should look into the chemicals that the cells are secreting and try to address inflammation first. So these steps are not well understood. My hope is that with 3D bioprinting we will be able to have better models which will resemble a little bit more what's going on in the real tissue. And here is just to show how the printer looks like. It's more or less like this big. You can attach to the head of the printer things such as cells or collagen or any kind of matrix, gel-like matrix, and then you can combine them as they were inks. Like in the regular printer, we have the different colors for printing the things. In the bioprinting, we can add different cells, different matrix, different components to try to build your tissue. So here, I have been working a little bit in collaboration with Dr. Adrian West from the physiology department, and those images are from him. So here are two examples, for example. That's how the cells would look like in just a regular model of regular culture, and that's how they are after they are printed in this kind of tissue. So we can see they look like a little more like cells than in the other image. And another big and interesting things from the engineering point of view, the mechanical engineering, is you can project some structures. So for example, this is a project that we have been working on in trying to mimic an airway. So the airway is a tube, and it's important to have the tubular structure as high as possible to try to have all those tensile forces driving the contraction, the expansion, and that's something we can try to do with 3D bioprinting. Otherwise, we just have the gel or the tissue. We don't have the freedom to try to build any more complex structures. We can actually change, for example, the red material there could be one thing, the blue part could be made of another thing, so we can play a little bit and feed different cells depending on the area. So we can try to really, I'm sure many of you have seen that there are people working on printing ears or, I don't know, pieces of skin, hearts and stuff like that. And that's just a little example of some of the latest experiments that I have done. We are testing different types of ink. So we are changing the composition of this matrix by changing a little bit the proportion between, let's say, collagen fragmenting and feeding those fibroblasts in there and seeing how they behave, if they are happier in one or another environment, if they are expressing this or that, depending on what we put in there as a ink. And yeah, so in a little less than a month, I will start my solo flights a little bit. Of course, I will still rely on my collaborators, but I'm very excited to move towards this direction. So we will have a bioprinting lab, and my plan is to combine all those optical or imaging techniques to visualize whatever we can get from the printer. The main question that I would like to answer in the short, medium term is related to how these fibroblasts are behaving as we move towards synthetic materials. So can we find something that will represent the extracellular matrix as nicely as the real environment in terms of the responses that they will have? Also, I would like to see how it's affecting the collagen remodeling in asthma. And I would like to see if we can optimize all these parameters to build structures, not only for asthma down the road, but also for other diseases and things like that. Yeah, so today, that's the end of my story. So I told you that nonlinear microscopy is a very powerful tool for imaging collagen and elastin. I told you also a cool example of application, showing how we can use that to drive some new conclusions in terms of the biological aspects of some diseases. In this case was asthma. It always started with those images showing us how the collagen was changing there. Then we went down all the way to the gene expression, protein expression, and we found something that has not been known by the lung community for many, many years. So everyone is really happy with us that a publication out of it in a very nice journal. So it seems that has been causing a big impact in the community. And I also mentioned a little bit of my future plans. So if any of you eventually plan to move to Ottawa to do a PhD, master's, don't feel shy to contact me. And finally, those are the people that have been involved in most of this work. My lab up to December 31st, the Hackett Lab, and the Center for Heart and Lung Innovation. We're still working with some people here at the MRC, Dr. Alex Cole. Adrian West at the physiology. If any of you would like to work with 3D bioprinting, contact him. He has a printer here in Manitoba. He's starting to do some very cool work, so it's a nice contact. And yeah, and all the funding agencies. Yeah, so thank you very much.
There's time for questions, yeah?
Yeah, so thank you for your presentation. I think you taught so marvelously well. Your work, especially the electricity part, it's so related to what I'm doing. So I think we have to discuss.
Nice.
But so looking at collagen, we know it has these plastic bands, and it varies from disease to disease. So did you check that?
It varies what?
Did you check if there's any differences within B-banding from cancerous cells, the collagen and the control? Did you check it?
For the cancer? No, I have not looked at cancer yet.
No, not cancer, sorry. Let's say cancer. Oh, my bad. I mean the asthma,
Yeah, no, so yeah, we saw some, we didn't see difference, for example, in the thickness of the fiber. That was the same for both. But for example, I have another work going on in parallel with COPD, that's lung fibrosis. In that case, the changes are really dramatic. We see basically, the work still, it's not published yet, so I don't know how much you can tell, but it's basically the fibers in the COPD patients are a little thinner and not as well structured as in healthy people or controls. Of course, it follows the patterns that we have less organization, everything in there, but in COPD we see a little more drastic changes in terms of the fiber itself.
Okay, question. Thank you for sharing your research. Just my question is, when you started the presentation, you provided a new imaging technique better than what has been done in ETs. But progressing down the presentation, we went back to staining the samples again. You went back to
I
staining.
was just showing the products.
Oh, so I was just trying to know, because prior,
Okay, yeah, in the
stains.
middle, when I showed the purple collagen.
Yes.
That was because we needed to know where some specific types of cells were in the tissue. So, and using the nonlinear microscopy, we only can do so much. So, for example, nonlinear is very good if you are interested in the matrix components. So, for example, elastin, collagen. But if you need to know the types of cells or things like that, then you need to stain them and use confocal microscopy, for example. Yeah, I'm not saying that these will completely kill staining. No, not at all. It just depends on what you're looking for. In our case, we wanted to see a little better details than matrix or the collagen. With staining, we couldn't see it. We just saw basically if there is more or less, but we couldn't see the fiber or anything like that. But it depends. If you're interested in cellular biology or things like that, for sure, you have to still staining. Yeah.
The microscopic, like photo taken from in vitro samples, right? My question is that how many cells did you like investigate to like do that comparison between control and
For which part? We have gels that were seeded with, if I'm not mistaken, 72,000 cells. And then we were looking at mosaics of these gels, about 2 millimeters by 2 millimeters, and then looking at each cell that was in that area. So that was about anything between 30 to 100 cells. It really depended on the culture. But then we would look individually at each one, and then average per donor per.
yeah, and like the photo that was taken.
That was a very high resolution one of the 2 cells.
No, I mean like, was it in the center of the culture, or how did you choose that 2 millimeter by 2 millimeter area?
It was actually what I did was we have that gel.
Mhm.
The gel is not that big. The gel is probably the size of your thumbnail. And we were doing 3 different locations, 2 by 2 each, in 3 different locations, randomly chosen. So it's a circle, so basically top and two.
Okay. Thank you.
Your gels, can you see through them with a light microscope?
Yeah, if you stain the cells, yeah, they are pretty much
No,
transparent.
not staining, just a plain white microscope, but then,
You could, but I'm not sure exactly what you get from those without staining. You wouldn't have to stain at least the cells, right, to have something.
I'm looking at something that is sort of self-staining, and
Mm.
I just wondered how well.
Yeah, for the collagen, let's say for the images of the collagen from the gels, in this case we did not stain. We just put it into this microscope. I don't think you would see anything if you go into a light microscope. You would probably see just a little blur.
Okay, so it's not very transparent for,
It's a little opaque. It's not super opaque, but it's not, yeah,
all right.
yeah.
I was kind of curious, just if there are no other questions, we still have a minute, I think. I think you were quite modest there. I looked up this journal because this is way outside my field, but it looks like a really good journal. And so I was wondering if sort of this new thing that you found in terms of the packing density in this one target that would control that, if this leads, and maybe you can't talk about this if you're collaborating with industry partners too much, but if this sort of has direct implications for therapeutics, like novel therapies, is this something that there are known treatments for, or things that could be tested for asthma?
That's the first step for asthma.
Yeah.
It has been shown that decorating affects always collagen packing in other models in knockout mice and so on. But for us, that was the first time that was shown.
Right.
And we still, it's interesting because we published this paper was in August, and we're still getting a lot of questions. We are at the phase that we are basically telling everybody that that's what's going on, not whatever we were thinking for. So the community is still curious about it, and we had some interest from the industry, but that was just the first step, and it's still basic science work.
Sure.
We have to increase.
But I guess my question was like, are there any known sort of things already that are known to upregulate or somehow change decorin or anything like that?
I'm afraid I'm not able to answer that, just
Okay.
because I'm not a cell biologist, so
Gotcha.
that could be a question for my colleagues.
Gotcha.
I don't want to say anything wrong here, so.
Fair enough. All right, well, in that case, thanks very much.